Department of Biotechnology and Bioinformatics at JP University of Information Technology. We are at Wakna Solar, Himachal Pradesh. My area of research is mainly related to environmental biotechnology. My research group is active in looking for some sustainable solutions of getting rid of the electronic waste. So we try to bioleach useful metals out of the waste, discarded electronic waste, with the help of microorganisms. Also, we look for conversion of the biodegradable waste, mainly lignocellulosic and the kitchen waste, to biogas and to biobriquettes. So, in all, we look for sustainable solutions for better environmental protection. Thank you.
Hello everyone, I am Dr. Anil Kant. I work as Associate Professor in the Department of Biotechnology and Bioinformatics at JP University, Wakna. I teach courses on molecular biology, recombinant DNA technology, genetic engineering, plant biotechnology and food fermentations. My research interests revolve around application of molecular biology in the area of plant biotechnology. I've done research on assessment of genetic diversity, molecular characterization of various plant resources using DNA-based markers and various other biotechnological techniques. Currently, I am working on development of RNAi-based solutions for better management of apple scale. I would be looking forward for any questions, queries on these topics. Thank you.
Okay. Hello dear students. I am Dr. Teesra Singh. I am faculty here in Department of Biotechnology and Bioinformatics. And you know this particular domain bioinformatics? What happened like few decades back, marriage happened between mother biology and father information sciences, and a beautiful child was born, and this child is named as bioinformatics. So if you want to make a wonderful career in this beautiful child related to this the domains which we are working on, you can choose this as a career, and it is one of the fast growing area, and you can see it has several extensions also, like you talk about the comparative computational and functional genomics, you talk about NGS data analysis, systems biology, synthetic biology, biomedical and healthcare informatics. All these domains are actually the bioinformatics subdomains. So if you want to analyze the data on computer, if you are tech savvy, IT savvy, then this domain is waiting for you to pursue an excellent career. So all the very best, and hope to see you soon in our university. Welcome again. Thank you.
Hello everyone, my name is Dr. Rahul Srivastav. I have done my PhD from Central Drug Research Institute, Lucknow. I am working as an Associate Professor at the Department of Biotechnology and Bioinformatics at JUIT Solon. My area of research interests are bacterial pathogenesis, antimicrobial resistance and biofilms. I conduct teaching and training in the field of immunotechnology, diagnostics and vaccines.
My name is Garlapati Vijay Kumar. I have done my PhD from IIT Kharagpur, and my research areas are bioprocess engineering, industrial biotechnology, and bioenergy and biofuels. And after my PhD, I have joined JUIT in 2010, and after that I went 2 years postdoc to Spain, and I am continuing as an associate professor now.
My name is Dr. Jagasin. So I am faculty here as an associate professor. So I work mostly with the genomics aspect of fungi. So in the biotechnology, we always depend on the technology. So there is always a continuous process of innovation. To boost those innovations, technologies are involved. So one of those technologies we are talking about here is the PCR. So we have in this lab the normal PCR that we use in the amplification of the DNA, and other one we have here is the real-time PCR, so that we all know in the COVID period. So we all have gone through that. So artificial, we do that. And on the basis of CT value, we calculate the fungal or the pathogen burden in the host itself. So these are the few things we have in this lab. And of course, more or less, technology is always evolving, and biotechnology is one of those streams which we see in the process of technology evolving. So that's what we have.
Hi, this is Dr. Hemant Sood, Associate Professor in Department of Biotechnology and Bioinformatics. I'm been with the JP University from 2009, and me and my team keeps on working on micropropagation of important plant species, which is lying in the northeastern Himalayas and or even in northwestern Himalayas. We are carrying out a micropropagation of these important plant species and trying to conserve it. We are also working on the production of important medicinal compounds by utilizing or by optimizing cell culture or tissue culture techniques for their production. So this is what we are doing with the Department of Biotechnology, JP University. Thank you so much.
Hello friends, I am Jiten Vasist. I am a doctorate from All India Institute of Medical Sciences, New Delhi. I am here in the Department of Biotechnology for 11 years. My specialization and working area is in infectious diseases, specifically for proteomics. We are working for the protein identification using proteomic technologies. Thank you.
I am Gopal Singh Best, and I did my PhD from Institute of Genomics and Integrative Biology, and the area is bioorganic chemistry. So after my PhD, I joined Ranbaxy Research Laboratory as a research scientist. So in Ranbaxy, we are responsible for drug synthesis and its optimization from lab scale to plant scale. And I am here in JP University since 2010. My current research area is antimicrobial peptides and peptidomatics. Basically, I do the synthesis of analogs of peptide and peptidomatics and we perform their biological evaluation, especially antimicrobial evaluation. And also, we are involved in extraction of bioactive material from medicinal plant. So these are my research area. Thank you very much.
Everyone, myself Dr. Poonam Sharma. I have done my PhD from Himachal Pradesh University Shimla in chemistry. My area of specialization is physical chemistry. My research is basically on thermodynamic properties, calculation of thermodynamic parameters with the use of surfactants as well as various drugs, right? I basically formulate various drugs formulations including new ingredients and also determining their CMC with the help of surfactants, right? For the surfactant determination CMCs, we basically calculate this conductivity with the help of this conductivity meter, right? And try to compensate with that of your CMC and calculate the thermodynamic parameters as delta H is there, delta S is there, and delta G, right? And then further, for the calculation of acoustical parameters like adiabatic compressibility, partial molar volumes, etc., we use this ultrasonic velocity meter. It is also called as ultrasonic interferometer for the determination of acoustical parameters. We determine velocity, sound velocity, along with that of the density with the help of this ultrasonic velocity meter for the calculation of various other acoustical parameters. And then for viscosity measurements, we use this Brookfield viscometer. This is your Brookfield viscometer. Basic principle is, and that depends upon its spinning of spindles, right? We apply torque, right? And with the torque, there is the rotation of spindle. That spindle further calculate the internal resistance between the layers of liquid. That is the basic principle of this Brookfield viscometer. So these are the basic instruments which are just used for calculating the physiochemical characteristics of various drug formulations. Thank you.
I am Dr. Uday Banu, Associate Professor in Department of Biotechnology and Bioinformatics in JP University of Information Technology. So I am actually teaching cell biology and then animal cell culture technology and also stem cell courses, right? So this is our lab. You can see here. So this is a laminar flow or biosafety cabinet, and this is a aseptic cabinet, you can say. And we handle all the animal cell lines inside this biosafety cabinet. And here you can see here, whatever cells we use, we observe under this microscope. So this is a special microscope, which is a inverted microscope. You can see here, the objective lenses are beneath the stage, right? So, and it has it is a fluorescent microscope also. We can observe fluorescent molecules, tags and probes, and we observe cell cultures on this microscope for observational purposes. And this incubator, carbon dioxide incubator, and we store the cell lines inside these incubators, right? So which maintains the carbon dioxide level, and also the humidity is also maintained in this. Right now we are not maintaining cell line, so whenever it is required we used to maintain it, right? So this is again another incubator. Yeah, here you have cell lines in it that is maintained. You can see here. So animal cell lines or human cell lines, they are maintained in media. So this is the culture flask that is used to culture the cell lines, right? And for observing these cell lines, we use those inverted microscope. And we hope any student who come to JP University, so they can utilize this facility, and hope to see you soon.
Hello guys, myself Dr. Saurabh Bansal. I am Assistant Professor at Department of Biotechnology and Bioinformatics at JUIT Vakna. I have done my PhD from Department of Biochemical Engineering and Biotechnology at IIT Delhi in 2012. Since then, I am here working as an Assistant Professor. I am here to teach you bioprocess engineering, downstream processing, industrial enzyme technologies, and protein engineering. My research focus area is over the identification of different enzymes, their purifications, their production and characterization, and the modification by using different protein engineering tools. Thank you.
So, myself, Dr. Abhishek Chaudhary, and I'm working here as Assistant Professor. I did my PhD from Indian Institute of Technology, Mumbai, and my Master's MTech from University Chennai. And my field of interest is nanobiotechnology. Basically, we are working for the development of various nanobiosensors and drug systems. Thank you.
Okay, hello everyone. My name is Dr. Ashok Nadda, and I am working as Assistant Professor Senior Grade in JP University of Information Technology in the Department of Biotechnology and Bioinformatics. Our area of specialization is microbial enzymes and conversion of CO2 into some valuable products. So using the microbial enzymes, we are purifying these and immobilizing these onto different nanostructures, different nano texture surfaces, and using these for the conversion of CO2 into some carbonates as well as some methanol or biofuels. We are working on the interdisciplinary field. We are also working on the microalgae cultivation and using these microalgae for the extraction of some value-added pigments and some biodiesel products. Okay, thank you very much.
Hello everyone. I am Dr. Rajkumar, working as an Assistant Professor in Department of Biotechnology and Bioinformatics.
I work in the fields of computer-aided drug design, structure-based drug design, and biomolecular simulations. Thank you.
I'm Dr. Shikha Mittal. Recently, I have joined JP University of Information Technology as an assistant professor. Before this, I was working as a project scientist in National Bureau of Plant Genetic Resources. My expertise is in next generation sequencing data analysis, in comparative genomics and genome-wide association studies, genomic selection, R language, and I'm also working in the field of Linux or programming.